It's my distinct pleasure to moderate this afternoon's panel discussion on the topic of disruptive innovation. As NC State celebrates its 125th anniversary, we spent a lot of time reflecting on our innovation and entrepreneurial history. From the development of food processing technologies to create shelf-stable foods, to the development of new drought and disease-tolerant plant varieties, to the launch of technology giants like SAS and Cree, NC State is truly changing the world. Our panel discussion this afternoon will focus on game-changing technologies that are being developed by companies that each have a presence here in North Carolina, which are bringing new benefits directly to consumers. The inventions they develop may create entirely new markets, or they may displace existing products and technologies. Great examples of disruptive innovation exist in things that we all will recognize: mass-produced automobiles, digital photography, and my favorite example, Wikipedia. So our distinguished panelists today don't need a lengthy introduction. Their bios are in your programs, but I'd like to welcome Dr. Al Bender, CEO of NanoVector, Dr. John Cavanagh, the founder and Chief Scientific Officer of Agile Sciences, and also William Neal Reynolds, Distinguished Professor in our Department of Biochemistry at NC State, Dr. Arno Krotzky, Vice President and Global Head of Crop Trait Knowledge at BASF Plant Science, and Dr. Michael Lassner, Vice President of Trait Technology, Trait Discovery and Technology at Pioneer, which is a DuPont company. So we're going to do this panel a little bit differently. I figured since it is the election season, we'd kind of make this like a debate. So I have a few questions that I'll pose to the panel. And we'll hopefully have an interactive discussion. We'll invite you to participate as an audience and ask questions throughout our session. So don't wait till the very end. We want to make sure that we address your questions as well about this topic of disruptive innovation. So for all of the panelists, the first question I'd like to throw out is to have you just talk about how you think disruptive innovation will continue to shape the economy, create jobs, and how your companies are actually encouraging your employees to be innovative and think outside of the box and develop these types of disruptive innovations. So Arno, why don't you start?
Yeah, thank you. Just realized here it seems that we are here more on the paper side. These guys are more on the IT side, you know, with their notes. So how do we encourage innovation in, at least in the company I've been working for now for, what, 25 years. I think it's fair to say that we have to recognize, and I mean a very good example for that is the Nobel Prize awarded today to a local university. I really would like to congratulate the incumbent that really game-changing innovations which are then carrying value. And innovation is not an invention. And innovation is something that carries value at the end, are done outside of the company. And we recognize this. And so we encourage our people whom we have to look outside as much as we can, as they can, connect to institutes, research institutes, and see what's coming. Because these innovations in our industry, life science industry, before they actually hit the road and actually generate value, as you know, take time. This is not, we're not going in product cycles in 2 years. So it's first of all looking outside. Second, looking inside. Innovations are not normally done by individuals. You have a combination of different people in a company, the ones who are, you know, connecting dots, you know, which I see, others who are specialists and so on. We try to encourage in our company to actually form innovation brain networks, if you want to call that, supported by IT systems. And we actually push this into the company by incentivization, by processes which we are guiding, because there's always, you know, the discussion that this is new, but then the question is it really innovative over time? So in that triangle, looking outside, scouting, be really, really radical, and also always come back to the baselines. What are we actually doing? try to feel out, work with the best partners in the industry. And I mean, I am lucky. I'm one of these guys. If you look at my CV, BASF 15 years ago had the idea to use metabolite profiling as a technology to look inside plants. This was at that time revolutionary. We started a company and we knew that it took 10 years to actually get up and actually looking inside the plants, build huge libraries of where we actually change genes in the plant, 75,000 by now, and looked inside the change, what the genes are actually doing inside the plant. That was at that time. It's now standard technology if you want. Still the companies are very big, the biggest companies in the world, but that was 15 years ago. And this is how BASF tends to operate. Look forward, look outside, build brain networks in the inside, build innovation platforms, and incentivize.
Great. Thanks, Arno. Al, I know you'll have a unique perspective as a serial entrepreneur and leading an NC State startup.
As I said, I'm leading NanoVector, an NC State startup. I think I'd focus on the economic impact of innovation. The technology, by definition, is innovative. We are using a plant virus as a component of a cancer drug, but we are farmers. We grow plants. I did some rough calculations. The value of this crop, if you were to grow it, is about $50,000 per pound. So the farmer, corn, virus, I mean, what do you want? In terms of, I think I'd pick the $50,000 a pound product. And then in terms of value per acre, I did another calculation, is that you can probably get a return of about $150,000 per acre of land for this crop. But there are some hooks to this. This has to be an FDA-approved drug, so you can't grow this in the ground outside. It has to be protected, so it requires greenhouses. And so there would be investment, but this is the new generation of farmers that farms biologics for drugs in greenhouses. And you also have to be able to be willing to withstand audits by the FDA to make sure you don't do anything that they're unhappy with. And so we're talking about a new generation of crops, a new generation of products, very high dollar products, but we also need a new generation of farmer that is willing to invest to create it.
So Mike, how is a giant company like DuPont and Pioneer also supporting an ecosystem related to disruptive innovation?
Yeah. So I guess I could start off by saying that Pioneer was really founded on disruptive innovation. So just, you know, in the 1920s when Pioneer was founded, the concept of a hybrid crop was completely disruptive because at the time farmers picked these big ears and saved them and planted them, and that's how they did breeding. And Henry Wallace, who founded Pioneer, actually had this concept of you get these scrawny inbreds, tiny little ears, that actually out-yielded what everyone else at the time was doing. And so Pioneer really has this culture of disruptive innovation. And then I think you can think about different kinds of disruptive technologies and how easy they are to adopt. And so because you know, our company was founded by scientists and is based on scientists, you know, kind of all the things about how do we do things better, cheaper, faster, make incremental improvements or maybe not so incremental improvements really are very easy to sell internally. So as new DNA sequencing technologies, new marker technologies came on, you know, those are really easy for people to grasp. And so, you know, just, you know, if you think back 20 years ago, we're just barely even using molecular markers, and then it was $20 a data point and $10 a data point. And, you know, just in the last few years, we've driven it down to like a nickel a data point. And so that means we're getting billions of data points and going to big data, you know, trying to understand what goes on a plant genetically, physiologically, all the signal transduction and transcriptional control networks. So that's, just huge thinking and innovation, both internally and externally. All the new tools about how are we going to discover more insect actives to stay ahead of the insects. It's really easy for us to get our head around anything we can do to parallelize, increase throughput, high throughput assays. Simple. The hard innovation is really thinking about how are we going to change the marketplace. So think about some of the innovation our industry has tried to bring forward where it's been really slow, maybe to the point of, almost not being successful, are changing grain quality. So, how can we make healthier grain? How can we make grain that has better feed efficiency? Technically fairly simple, but that requires kind of marketing innovation. And it's really challenging to think how you fit those kind of changes into a commodity system that's designed to resist change. Nonetheless, we really encourage that. And so that's kind of where you see the thinking about how we'll do the next generation of drought products. Maybe I'll stop there.
So John, as a faculty entrepreneur, why don't you share with the audience your experience with translating an early stage disruptive innovation to the market?
This is actually quite an interesting question. How do we innovate, or how do we promote innovation at a small company like Agile? So Agile is clearly not as big as BASF and DuPont, if you didn't know, and we have fairly fewer employees. And what we found, and we're a 5-year-old company, so we've ridden out some tough fiscal times. And one of the ways I think we've been able to do that and bring this very new technology that we've developed so far is, and I think this may be a decent model for other startups at this time, is that the company, most of the innovation I think is still done in the academic labs of Christian Melander, the other co-founder, and mine. And what the company has been very good at doing is leaving us alone. And I think that's been an enormously powerful thing that we're involved in the company intimately, of course, but we've been able to be very flexible in where we're looking and what work we're doing. And we're never really seduced too much by what's going on at the company. Of course, we pass our information to the company, but that ability to do what we want has actually driven our innovation very rapidly and much faster than in other places. We've dealt with some companies that move at a lot slower pace, and we're much more agile. But I think that's because there's been this beautiful partnership in the sense that we don't partner all the time. We're left up to our own devices. And my job, I'm an academic. My job is to be disruptive at work. I'm a disruptive element. I'm supposed to think of new things, as Christian is. We're not held to any particular strategy. And that, for us, I think, has worked wonders. So the fact to leave the scientists alone sometime that are developing the technology is a beautiful thing.
Arno and Mike, so can you tell us a bit about how you use information you gather from? All right, there we go. So Arno and Mike, can you talk a bit about how information you gather from supply chains inform your R&D strategy and also play into your thought process related to disruptive innovation? Mike, do you want to start?
Okay. Well, since you used the supply chain word, to me that means how do we produce seed, how do we manufacture it, and how we sell it. So I have a great example of that. We heard in the last talk about a biotech trait where you use biotech to propagate the crop, but the GM trait is not actually in the final product. And so one process that we've developed that we're particularly proud of is what we call our SPT hybrid corn production system. The way corn seed is produced these days is you have separate male and female plants, and the plants we harvest the hybrid seed off of are detasseled. And so for many years, we hire high school students in the summer. They go through and they whack the tops of the male flowers off the corn plants. And then we have rows of male plants interspersed, and we use them to pollinate the female rows, and that's where we harvest the seed we sell to our customers. And you know, this, as you can imagine, if you're a plant sitting in a cornfield in the middle of the summer, getting your top whacked off wouldn't be the most pleasant thing for you. So it reduces the yield, makes them more susceptible to disease. And so we developed this new process where we use a genetic male steril, and we have a transgenic maintainer. So this restores fertility to that. So that allows us to keep producing these plants over and over, but they're male sterile. They don't have a fertile male flower. And so what we've done is this transgenic restorer is attached to a pollen ablation gene, so that the restorer is in the plant, but it cannot be transmitted through the pollen. Pretty slick trick, and probably without diagramming it, if you don't understand plant reproductive biology, you know, you don't get what I just said. But what this means is we have a way to maintain sterile plants. We have much higher seed purity out of the process. We get much better seed yield. So our supply chain loves it because, you know, this reduces our cost of goods as well as increasing quality. Now, I think we heard at lunch, you know, okay, biotech brings incremental value to, you know, to U.S. farmers, but it's really a bigger deal in the third world. And so the analogy I'd make to what we're doing here is the next crop we're bringing this to is rice. Now, rice mostly is not hybrid, except in China, and there the government actually forced it on the system because hybrid rice really, at least when it first started, really didn't taste very good, but they wanted food security to increase yield of rice. So China is really the only country in the world that has a lot of hybrid rice. As a result, they have very high rice yields. One of the disadvantages of the system they use is around 75% or more, depending on if you're on the male or the female side, of genetic diversity available to breeders is not compatible with the hybrid rice production system we use, or that they use. Our system that we developed for corn is completely compatible with all different rice genetics. And so we're well on the way of our next, you know, kind of growth platform is hybrid rice in Asia. And so we've been bringing that technology over to rice. We now have, with our Chinese partner, we're in regulatory trials in China for the first year, in India, where we're developing this on our own. We also have regulatory trials going on to move this technology. We have to then get the breeders to change the whole base of the germplasm so that they can improve the genetic diversity base, bring higher yield and yield stability that you'd normally associate with a hybrid crop. So that's great. That's, you know, if the regulators let us go, we'll have that kind of around the end of the decade. But what's next? We heard about wheat not having technology applied to it, and wheat farmers in the U.S., you know, not benefiting it and actually coming back. That's kind of the next place we're going. We're just getting engaged in that. Wheat doesn't have hybrids. Wheat has very narrow genetics, and so we view this as the way to change the future of wheat.
Okay, I will look at this from a slightly different perspective. The reason is quite simple, because BASF Plant Science has a very unusual business model. Our business model is that we are partnering with leading partners in the breeding and the seed industry to bring our innovations to the market. So how do we actually then get your phrase, supply chain information in? Obviously BASF, we are part of BASF. BASF is a leader in crop protection and in functional crop care. So together with our partner, our own intelligence which we have, so we get a very good picture of what's actually needed in the market. But then I open our research supply chain, to the other side, and we are engaging in quality traits. For example, we are developing, and actually pretty fast here now, after some years of base research, bringing polyunsaturated fatty acids in canola to the market, where we are world leaders. We have 10 genes in there to enable this technology. We are working on feed, and far and foremost, which is by a long shot the most important area for us, is yield enhancement. Now, yield enhancement, it's quite easy to say, and I wouldn't say that it's easy to do this with a plant with GM. Why? Because you actually need to understand what the plant is doing. And so since we know what the market is, and we are working this together with a very big partner, as you know, for many years already, we have developed an incredibly good discovery and optimization platform, which is, I would say, world-leading, which allows us now to look inside the plant. And I can tell you it's not only the genes which tell you the story. It's much, much more. The proteins, metabolites, the pathways, which it's like a sensoric system. If you want to, take a, I don't want to make advertisement, but if you take a Chevy Malibu and want to take it and turn it into a Trans Am car, it's not enough to put a spoiler on it, or measure on the outside sort of how fast that car goes. You need to understand how that engine works, the gearbox works, which functions do the wheels have, that you don't put a mat in the car and hope that the car goes faster. So we have developed a platform which I'm quite proud of, which is so unique and so fast, which allows us now to look inside the plants when we actually start altering the plants by GM, and actually try then to understand how these genes are actually functioning, that we're becoming better and better and better in this optimization. So this, to me, is at the edge of I would say, disruptive technology, because it takes discovery by testing, by trying, to discovery by knowledge. And this is basically the same what you see in pharma. That's the same what you see right now in medical research, that we try to understand how the entities are actually working, like an engineer. And we've coined a new phrase. In our case, we are going to a next-gen systems biology. You heard that before. So we are looking into the plant as a machine, if I dare so, and actually try then to be very cognitive in optimization, then to meet the specifications which we get from the market.
Great. Thank you. So John, from a startup company perspective, do you think the venture capital community's insistence on being very focused kind of hinders the disruptive innovation cycle?
This is a great question for me.
That's why I chose you to answer the question.
I live in a very bipolar world. I was just noticing it says Agile Sciences on here. It says NC State on here. So you'll get a bipolar answer to this. Yes and no. I can definitively say that. It starts initially wearing my academic hat. Okay. I really worry about this being too focused. Very early on in the company's history, before our current management team was in place, and it was really being driven by a couple of very naive scientists, myself and Christian, we thought we were going to be a paint company. Now we're in agriculture and we're in human health. But we were going to put our molecules into paint, and we're going to solve all the paint problems ever known to man. And we talked to some people that had money that were very interested in moving in that direction. And you talk to a couple of scientists and academics, and any amount of money sounds like a lot of money because we have no money. And so we were all very excited about this. And it turns out that our molecules don't do that great in household paints, a little bit better in the marine area. But Had we taken that money and been focused on that, which the VCs at that time were very insistent on, we would probably not be sitting here right now. We would not have had any flexibility. We would have focused very much on that one area, and we would have lost out. Now, we've been given, so we muddled through ourselves, and we were very flexible. We are able to now define the correct areas we should be in, and now we're very focused in those areas. And now, we're ready to, if anybody has any money out there, hi. We're actually at the point where we're looking for money, but now we're in the position where we're looking in the right places. Had we done it earlier, no, it would have been a death sentence for us, I think. And I think it's all about timing, certainly for startups. A lot of my friends on the West Coast started biotech companies, and every single one of them went out of business, and that was either from taking money too soon or from the scientific founders becoming overly involved in the business side. But I think you can be careful what you wish for. You can get the money and it can put you out of business if you're not careful.
So Al, I can see you're ready to jump out of your seat. You've raised a lot of money for small companies. What is your...
Well,
that was for our particular company.
In terms of the focus, I've been involved with venture capital for 25 years, and they've always want you to focus. It's, in fact, they'll say focus, focus, focus. But they also realize very few companies ever end up producing what they originally invested in. And it may come as a surprise to this audience, since I'm a student of it, that Google came within $1 million of shutting down its business. That's how struggled they were. And they had a board meeting with a decision, should we close this business down? And they asked one of the technologists, founders, do you have any new ideas? And they said, well, we have this idea for pay-per-click. And said, well, we'll go try that. And so they did something different, even though we're focusing, and they are to the Google that we know today. And I can tell you that pretty much every company goes through that phase where they have to change direction. But even once they change direction, though, they stayed focused on the direction that they changed.
Well, we've been told flat out, you focus, don't do anything else.
Absolutely.
And we would, trying to jump off and do anything that's slightly at a tangent to that would have put the final nail in the coffin, I think. I think the fact that we were, and certainly and this is in our case, that we were wise enough to work out the kinks ourselves make us a lot more palatable for people now, that we're focused on the right things now. I think that's, in my, you know infinitely more about this than I do. I absolutely hold my hands up.
But then they,
but in our case, we were too messy to start with. We were focusing on the wrong things. We needed our time to grow and breathe and evolve at our natural rate before we become attractive. So I would hate to have taken money too soon.
And that's absolutely correct. You have to focus, but you also have to have a plan B if plan A goes south.
I'm good at that. My bipolar world clearly is. I got plan F, G.
Arno, you had some comments?
Yeah, I just want to add to something to that. BASF came into the race for plant biotech, as you most likely know, relatively late. Actually, about 10 years later than most of the other companies. So we adopted a strategy. It's not only like venture capital companies, that BASF said, where are the innovative centers in the world, and can we actually form companies with them and start? And this is what we actually did. But so I led 2 of these companies over the last 14 or 15 years, now 14 years basically. So there's a model where you have these really gigantic companies who are learning also to work like VCs, but not, believe me, we were focused. We had to be focused. But you're right, we had to have plan B, and in many cases also plan C. Yeah, I had to change these companies. But it's a very viable system, actually, to catch up to competition in relatively short time, because you're not basically burdened, if I may say, of large company decision processes. You can be much, much more agile like in the real world, but then you have an investor like BASF, which has the understanding that something like a plant biotech branch, you're not setting up in 3 years. So even in the board at BASF, you have guys who came from research, and they know that it takes longer, but still focus had to be brutally, I can tell you, yeah.
Do we have questions from the audience?
I thought it was a good idea to sit all the way in the back. I realized it wrong. If you want to ask the question, it's like a march. My name is Justin Lee, work for BASF Plant Science. I have some questions for the panels. What do you think would be the characteristics for disruptive technology in biotech, specifically in ag biotech? Mike, you mentioned that hybrid being the disruptive technology in the 20s. So is yield the only factor? Because it improves yield, that's what you said. And then I would like to hear, just for my own curiosity, would you consider RNAi technology a disruptive technology for the upcoming future? Thanks.
So first off, I'd say hybrid vigor is more than about yield. It's about yield stability. It's about genetic diversity. I think we heard at lunch that one thing it did is it actually gave some intellectual property protection to the product, because farmers couldn't save their own seed. They have to, if they save their own seed, they don't get the benefit of the breeding. So what it does is it gives companies an incentive to invest in breeding and crop improvement. And that's at least as important as the hybrid vigor. I think there are some future systems, and so that's really about changing the rate of genetic gain. And so, we heard a lot today about how are we going to, feed everybody without that much more land, and what it's going to require is these investments in stability over environments, to climate change, to investment in increasing the rate of genetic gain every year, much faster than we ever have in the past. And then I guess I'll jump to RNAi. Actually, RNAi is a very old technology. I used to work at a company called Calgene, and we brought the Flavor Saver tomato on the market. This was actually labeled by choice. Our brand was McGregor's tomato. We tried to make it sound like McGregor Farms. And we thought it was antisense at the time because we were expressing the gene backwards. Actually, now that we understand it better, it was RNAi. The high oleic soybean that we're just waiting for EU approval before we introduce it more broadly is RNAi technology. And Monsanto has a similar product that they're in the regulatory process with. That's RNAi technology. So it's not really new. It's very powerful, and it's just one of the tools we have in our toolbox about how can we control genes, putting them up or putting them down. RNA interference. So essentially what you're doing is you're using the plant's defense system to control viruses to turn off endogenous genes.
Time for one more question.
Good afternoon. My name is Easton Horner. I'm a research scientist at Syngenta. And I was just curious, as a research scientist, how do you balance destructive technologies, new ideas, with the conflicting point of view sometimes of optimization of existing ideas to be better and better? How do you balance disrupting your optimization, and your thoughts on those?
There's There's a lot of science around that, and usually very large companies are very poor at disruptive technologies, and the smart companies figure out how to do it. Some of the examples are that the leader in the credit card business in the early days was Sears and Roebuck, but they didn't know what they had because they couldn't see it for what it might possible business. So the way to bring big companies can adopt disruptive technologies is they usually take a group of people and they move them aside, maybe in a separate operations, separate locations, give them a vision and give them some resources and let them just go at it. And that is how the most successful companies, and if you want to read some books on it, there is Crossing the Chasm and several others that talk about that topic of how to develop, how to bring destructive or advanced technologies into corporations that are consumed with having to solve the day-to-day problems of their customers, which are typically incremental solutions, not destructive.
Yeah, I can just add to this, and I think you're totally right. At least in the company I come from, BASF, global, we have, 110,000 people working. We call them so-called growth clusters. These are people, yeah, who are sort of outside of the, I would say, of the mainstream, trying to achieve something which is very early. Sometimes they do think out of the box, and it's like with the VC company, 80% of that stuff goes down the drain because you cannot achieve it. A good example that some things can also happen, it's not plant biotech, obviously BASF Plant Science, my company, has started like this 10 years ago, but there are other growth classes like in BASF, like when they developed the organic LED. Yeah, that was something where people thought that would not be achievable. 10 years later, they finally did it, but it was like, you know, you're flying very low, and if that wouldn't have happened, you know, the money would have been pulled. So it's exactly that. You have separated rooms of, you know, innovation thinking, out-of-the-box thinking, and there is usually sufficient time to actually test that. But it's not always a guarantee that it works. That's quite clear.
I'm going to chime in just a little on this, I think. So my understanding is, you know, when do you focus on disruptive novel innovations and when do you try and optimize existing ones? And I'm talking about this just from our company's point of view, so it may not mean anything to anybody else. We've managed to avoid that, I think. I've never really thought about this. In our case, we're actually taking advantage of the fact that you can't optimize the things that we're trying to replace or help out. Our technology is, nothing works, right? Nothing works in human health or ag health. We've got resistance all over the place, fungal resistance, bacterial resistance. So it's really difficult to treat disease, period. And so optimizing current ways of doing things, it's impossible. You know, your systems that you're trying to fight are a lot smarter than you are. We've managed to avoid that optimization issue by going completely away from it, and our technology doesn't worry about that. It's brand new. We go in and we destroy protection systems from a very different vantage point. So to my mind, if you're really doing something completely novel, and in our case, I think our technology is completely novel, we've not had to worry about balancing between optimization of existing technologies. We're just doing something.
Just a quick comment, Al.
Just one more last comment. I just remembered a great book is The Innovator's Dilemma. I forgot that book, which addresses that topic exactly, and highly recommend that anyone interested read it.
So thank you, everyone. I think we're out of time. Please join me in thanking our panel.